King. So let me first introduce myself. I'm Ceri Lewis. I'm a marine biologist. So I probably think about fibres in quite a different way to most of the people involved in the PCT and these interesting networks. I've been studying the impact of changes in our ocean on a range of marine animals. I generally focus on the smaller invertebrates that live the bottom of the food chain, and looking at things like climate change and pollution impacts on these environments, these species, for most of my career. And obviously, one of the big pollutants that we're finding in the world's oceans at the moment are plastics. We find an awful lot of plastics in our ocean. The numbers are big. So on my screen now is just the latest kind of global estimate of how many plastic particles are floating in the surface of the ocean. This is just the surface. So 170 trillion plastic particles. And we know that it's not just on the surface of the ocean. There's plastic throughout the whole ocean, even the deepest parts of our ocean. So this is something we've been working on for about 17 years now. Myself and Tamara started working on this quite a long time ago when we were both at Plymouth, before we even moved to Exeter. But when we started working on plastic pollution, we were focusing much more on these particles that you can see in this image here, tiny microplastical particles, things that have been broken down from larger fragments. But we were finding fibres in our samples, but quite often we were ignoring them because we weren't sure if we were sampling them in a quantitative way. They generally pass through a lot of the nets we would use for sampling particles. We weren't always sure if they were plastic or not. And so an awful lot of the early research into plastic pollution was kind of ignoring, dismissing, even, deliberately excluding fibres from our data. Then we realised that fibres were regularly appearing in these samples, regularly appearing in the species we were looking at. And so actually we've started to pay much more attention to this fibre contamination, as have many other researchers working on this problem around the world. And so we're now starting to realise just how widespread fibre pollution is in our oceans, as well as these other microplastic particles. This is just one of the few global analyses. There's not many global analyses because of this problem of lacking data. But here you can see that there's fibre pollution. Basically, you're looking at the track of a ship that's done a global cruise and sampled particles in the oceans. And they've gone around Antarctica. So we're even finding microfibre pollution in some of the most remote parts of our ocean, in the Southern Ocean, miles away from any big cities. But generally, we find them really high abundances in the more coastal areas of our ocean, closer to wastewater effluents, for example, or coastal inputs of plastics. So we've been studying how these get taken up into, as I said, animals at the bottom of the marine food web. So mussels are a really useful species to be looking at these sorts of questions with. You might look at this photo and think of a nice dinner with some chips. I look at this photo and think something really quite different, because I think of them as sort of passive samplers. Mussels feed by filtering huge volumes of water over these little sort of frilly structures, which are their feeding apparatus. And you can see in this time lapse of some mussels in our lab, the water is getting clearer because they're filtering the algae out of the water. I'll play that again. So you can see the water starts really green and then it's going clear because they're filtering all of that water over these structures, pulling out the food, which is the green algae in here. You can also see these kind of little pseudo feces being spat out at the bottom. So some of that is going into the mussel and some of it's being filtered and then passed out as these kind of pseudo feces. And so they'll take up anything that's in the water. It's about 8 litres an hour that one mussel will process. And so they're taking up any kind of pollutant in the water, including, as it turns out, plastic particles. So we did a study. We went to a whole range of different sites around the southwest of the UK and just looked to see what plastics we could find inside all of these mussels. And what you're looking at in this graph is that we found plastics everywhere. In I think it was 89% of the actual individual mussels we looked at. But if we describe the shape of that plastic particle, the majority of them are fibres. And here we've just counted the microplastic fibres. So we excluded any that didn't passed our FTIR test as being a petroleum-based fibre in this particular study. But that's something that we've now started thinking about changing, because we know that there's a whole range of other fibres that we find as well. So our thinking has already changed since we did the study in 2019 about how we should be reporting these fibres. And then we've been invited to Galapagos. And the reason that we've been working in Galapagos is firstly because we were invited by a really passionate researcher who was actually a turtle researcher, who started finding plastics all over the beaches while he was doing his turtle conservation research. And he wanted some help mapping plastics in Galapagos. But for us, Galapagos is a really fascinating case study because it's this remote island system. It's far away from any of the big oceanic gyres, which is generally where microplastics floating on the surface ocean would concentrate, and yet it's experiencing these high levels of pollution. But obviously Galapagos is famous for its really unique wildlife. It was the inspiration for Darwin's theory of evolution. These beautiful blue-footed boobies doing their little mating dance are one of the iconic species, but it also is one of the richest marine habitats. The oceans are absolutely full of wildlife. It's also the sharkiest part of the world's oceans, which is a really positive sign of a healthy functioning ecosystem to be able to support that many top predators. I have been lucky enough to go diving with the hammerheads. It is totally awesome. And you can see in this photo of these beautiful sea lions and other iconic species of the Galapagos how dark and green the water is that these sea lions are playing in. And they're also very playful and like to try and consume plastics through slightly unusual routes by chewing on our fins. But the reason the water is so dark is because it's full of plankton. It's a really nutrient-rich environment. And it's these currents that come from the Southern Ocean, and they come up the coast of Latin America and then spill out across the equator. So Galapagos is situated on the equator, about 1,000 kilometers off the coast of Ecuador. And it's based in these cold water currents that have come all the way up from the Southern Ocean. And Galapagos is volcanic islands, so they're really steeply sided. So this cold water comes up and then shoots up to the surface, hits that equatorial sunshine. So it's nutrient rich, hits the sunshine and just explodes into this abundance of life. And that's what makes it such a rich, unique ecosystem for supporting sharks and all these beautiful wildlife. But it's these currents that also bring with it the waste from mainland Ecuador. So it's being bathed in plastic litter coming in on these currents from the mismanaged waste, unfortunately, on Ecuador and Peru, where they don't have particularly good waste management systems at the moment. And an awful lot of rubbish basically falls into the sea, gets transported across on those ocean currents. And so that's why we've been studying it, because it's just such a fascinating system, but it's also a closed system. Everything, because Galapagos is so protected because of its unique wildlife, anything that's being shipped into the islands to support the tourism industry there, or the locals that live there, and locals do live in Galapagos. There's about 30,000 people living in very controlled areas on 3 of the islands. But it's all really well documented, and so we can actually map the flows of plastic. We can map what's coming in and being used locally versus what's coming on these ocean currents, and really start to understand the sort of flows of plastic pollution in this system. And here's just some of the photos of what it looked like the very first time we went to Galapagos. When you go as a tourist, you get taken to one side of the island, which is where all the beautiful sandy beaches are, which is the more sheltered side of the island, because it's easier to land there. Off the boats, you're normally visiting via boats, whereas if you go around to the other side of the island, facing those currents and those prevailing currents from mainland Latin America, the first time we went, about 5 years ago now, this is what we were greeted with, because nobody else was really going there, nobody was clearing it up. And then you can see this is actually coral rubble, which was the coral died from an El Nino event, which made it too hot. But then there's an awful lot of plastic now collected in that coral rubble. And so we've been doing all sorts of sampling in Galapagos, looking at what's in the water, what's in the sediment at the bottom, what's in some of those the invertebrates that live there. We don't go sampling sea lions, you'll be pleased to hear, but we are looking at some of the invertebrates again at the bottom of the food chain, trying to understand how this plastic pollution is behaving, where it's coming from, what we might be able to do about it. And again, you can see in this image here, so this is the end of a plankton net. When we trawl it through the water, it collects all the particles. And yes, we can see the fragments, which probably come from large items, but there's an awful lot of fibres in there as well. And so we've spent a lot of time trying to make sense of the particles that we find, and we've realised that actually fibres form a really large percentage of these microplastic particles when we're sampling nearer the urban areas of Galapagos. So when we're on those remote sites that aren't really visited, there's nobody living nearby, it's mostly the larger particles that we find. But when we sample in the harbour and around the small town where there is a wastewater treatment, well, I say treatment, there's a wastewater pipe. We're not really sure how much treatment goes on. We suspect an awful lot of what comes out of people's washing machines is probably going straight into the sea. And so we've started to get a better sense of the fibre contamination, which may or may not be from local sources. It's actually really hard to tell. But what I'm hoping you get from looking at this figure is firstly the numbers. So 12,300 particles per metre cubed in Galapagos seawater around this harbour, and an awful lot of it is these what we would call anthropogenic cellulose fibres. So an awful lot of it is polyester, but a good proportion of it is what we call anthropogenic cellulose fibres. We're finding it really difficult to unpick this any further, and that's where joining this network has become really, really useful for us, because we're now learning much more about different methods we can use for looking at these fibres to get a better sense of maybe what sort of textiles or rope that they came from, how we can make more sense of this data, because our sort of limited FTIR expertise generally just gives us really crude material types. When I say material, I mean like it'll tell us it will be polyester or polyamide or anthropogenic cellulose, and doesn't really tell us much more about it. But we're finding lots of them. Here's just a few more photos of these. So these have been fished out of the sea. Sometimes we find them in the guts of marine animals. So these are the sorts of animals we were looking at. We weren't sampling the Sally Lightfoot crabs, it was actually the barnacles that we're sampling. Again, they feed with this kind of filtering action, which means they're capturing lots of these particles. Sea urchins. This is actually a fragment from a larger plastic tub. You can even see the tiny little teeth marks from the urchin that's grazed those, and that was taken from the gut of a sea urchin. But an awful lot of these fibres, some of them look like fishing gear, an awful lot of them don't, and they look more like textile fibres. But again, our ability to differentiate at the moment is not great, and that's really what brought us to this project. Even our iconic marine iguanas that live in Galapagos are ingesting these microfibers. So again, we weren't actually studying the iguanas themselves. We were collecting their fecal matter. Turns out if you pick up an iguana and stick it in a plastic tub, it pretty much poos on demand for you, which is quite useful, so we can get uncontaminated samples. We had a vet, and it was all done with permits and Galapagos Park rangers with us, but that's told us that actually an awful lot of these marine iguanas are also ingesting these microfibers, and it's probably because it's tangling up in their food. So they scrape algae off the rocks, and we think these plastic contaminants are just getting trapped in the algae, and then they're ingesting them by accident as they're feeding. So a very, very cool, iconic species. I'm pretty sure Godzilla was based on one of these. Look at them, they're such dudes. But they're also ingesting these microfibers. And, it's not just Galapagos. We find this in pretty much every species that we look at. So we have data from other colleagues in Exeter who've looked at, cetaceans around the UK. This is all done with strandings. But whenever an animal a necropsy is washed up, it will be investigated. So all of this data comes from strandings, but there was an awful lot of plastics within the guts of all the dolphins that were studied. Turtles, 100% of turtles studied through necropsy research have been found to have microplastics in their gut. 70% of UK shark species have been found to contain microfibers within their guts. And as I said, even in the deepest part of the world's oceans, bottom of the Mariana Trench, people are finding microfibers of various types within the guts of marine animals. So this is an increasingly recognised problem now. And so we're really interested in learning more about, firstly, how these fibres are actually impacting the health of these animals, and then by understanding a bit more about which fibres are coming from where, what we might be able to do to mitigate this form of pollution. So we have done some studies on what ingesting particles means for the health of these animals, but an awful lot of the research that's been done on this for plastics has been done on those microbeads, the more sort of round particles. And round particles pass through guts relatively easily. Whereas actually what we're finding when we go and collect species from the wild is that, they're containing many more fibres than these particles. We have done some studies we've got a research aquarium where we can do controlled exposures to understand these impacts. We've done some work with crabs, and crabs actually have quite interesting physiology. They actually breathe through their legs. You might not have realised. They draw water current up through their legs, it then passes over their gills and then goes out through their mouth. So it looks like they're blowing bubbles at you. It's very cute. And so they've got these quite complicated gill structures, which basically trap any fibres. They get caught on that structure. And then anything that's being accidentally ingested when they eat it with their food, they have a gastric mill, which again is quite a kind of complicated physiological structure, and the fibres get caught on that. So crabs will actually retain microfibres for months at a time compared to those beads, the more kind of traditionally used type of plastic for these experiments, that will pass through their guts quite easily. We've also done some work on things like copepods, trying to understand what happens when they're ingesting plastics. And a pattern that has been repeated in many different experiments, not just in our lab group, but labs all around the world that looked at this, is that when animals that are small with high turnover, high metabolism, which most small marine invertebrates do have, when they're ingesting plastics, they're ingesting less of their natural food because the plastic's getting in the way, it's taking space in their guts, so they're getting less energy through their diet. And because their basal metabolic rate is going to stay the same, that means there's less energy left for reproduction, which obviously contributes to their fitness, and then egestion, which contributes to things like carbon fluxes in the ocean. How much of copepod poos influences how much carbon is moved from the surface of the ocean to the deep ocean. So there's some potentially quite big impacts of eating plastic for these animals. But this is something that we really want to understand much more carefully, and particularly for fibres and not just plastic fibres. So let me introduce Holly. I think Holly is on the call. Holly is our relatively new PhD student who's joined as part of this Fibres network, but also through our Centre for Resilience, Environment, Water and Waste at Exeter, which is a collaboration with South West Water. And Holly's going to be looking much more in depth at these fibres. Firstly, we're going to be looking, we're going back to Galapagos in March. We're going to be collecting more of those fibres to see if we can get a better sense of what type of fibres there are. And that's why we're really tapping into the expertise of this network to help us look at these a bit more carefully and see if we can learn a bit more about what items they might be coming from. Is it coming from people's washing machines? Can we look at particular items? Because if it is washing machine waste, there's probably things we can do to stop that getting in the ocean, but we probably need a bit more evidence to support that first. And then Holly's also going to be doing some ecotoxicology experiments to try and get better data on the impacts of ingesting these fibres, but also not just looking at the plastic fibres, looking at a range of other textile fibres, because we really want to know if natural fibres are actually better or if they're no different in terms of the impacts once they're ingested by something like a mussel or a crab. And we've got quite a cool experiment planned to look at this. So this is a system in our research aquarium where we can measure real time the uptake and responses of mussels ingesting these particles. So we fit our mussels with these sensors. We can measure the mussel's heart rate, the same way, our heart rate shows how healthy we are. We can kind of do the same with mussels. We can measure their gape. So we've got some little basically magnets attached that will tell when the mussel is open or closed, which help us understand whether it's responding to fibres coming into its water by closing or whether it can't tell and it's feeding normally. Sometimes they can do what we call a coughing or flapping response to kind of cough particles out. So we're going to be looking at that in more detail. And then we can measure their respiration rate. So sorts of things you might have measured in you if you went to the doctors feeling a bit poorly. And then expose them to a range of different fibres, measure real-time responses, and then look at things like oxidative stress, DNA damage, to see how they're responding to that exposure. And obviously, our motivation for doing all of this work is to better protect our marine animals that we know are ingesting these in all parts of the ocean, but particularly we want to keep our Galapagos species happy because they are so special. Thank you very much. I will stop sharing my screen. Oh, and I forgot to say, for Holly's experiments, we are desperate for fibres. So if anyone has a sack of fibres sat in their office, we would love them. But we'll be working with partners across the network to get fibres from various different sources.